Section 3.4 Proteins. Proteins, as their Greek derivation implies, are of primary importance to the structure and functions of cells. As much as 50% of the dry weight of most cells consist of proteins. Presently, over 100,000 proteins have been identified. Here are some of their many functions in animals. Metabolism. Enzymes bring reactants together, thereby speed chemical reactions in cells. They are specific for one or more particular type of reaction that can function at body temperature. Support. Some proteins have a structural function. For example, keratin makes up hair and nails, while collagen lends support to ligaments, tendons, and skin. Transport. Channel and carrier proteins in the plasma membrane allow substances to enter and exit cells. Some other proteins transport molecules in the blood of animals. Hemoglobin is the complex protein that transports oxygen. Defense. Antibodies are proteins that combine with foreign substances called antigens. In this way, they prevent antigens from destroying cells and upsetting homeostasis. Regulation. Hormones by regulatory proteins. They serve as intracellular messengers that influence the metabolism of cells. The hormone insulin regulates the content of glucose in the blood and in the cells. The presence of the growth of the hormone determines the height of the individual. Motion. The contractile positions, actin and myelin, allow parts of the cell to move and cause muscles to contract. Muscle contraction accounts for the movement of animals from place to place. All cells contain protein that allows cell components to move from place to place. Without such proteins cells would not be able to function. Proteins are such a major part of living organisms that tissues and cells of the body can sometimes be characterized by the proteins they can contain or produce. For example, muscle cells contain large amounts of actin and myosin for contraction. Red blood cells and cells that are filled with hemoglobin for oxygen support and support tissues such as ligaments, tendons contain fiber, protein, and collagen, which is composed of tough fibers. Peptides. Proteins are polymers with amino acid monomers. Figure 3.15 shows how two amino acids join by dehydration reaction between the carboxyl group one and the amino group to the other. The resulting covalent bond between the two amino acids is a peptide bond. These atoms associated with the peptide bond share the electrons unevenly. Because oxygen is more electronegative than nitrogen, they force the hydrogen to attach to nitrogen has a slightly positive charge, while the oxygen has a slightly negative charge. The polarity of the peptide bonds means that hydrogen bonding is possible between the CO of one amino acid and the NH of another amino acid in a peptide. The peptide is two or more of the amino acids bonded together in a peptide. This is a chain of many amino acids joined together by peptide bonds. A protein may contain more than one peptide chain. Therefore, you can see why a protein can have a very large number of amino acids. In 1953, Frederick Sanger developed a method to determine the sequence of amino acids in the peptide. Now that we know the sequence of 1,000 polypeptides, it's clear that each polypeptide is its own normal sequence. This sequence influences the final three-dimensional shapes of the protein. Proteins that have an abnormal sequence have the wrong shape and cannot function properly. Figure 3.15, synthesis and degradation of a peptide. Following the dehydration reaction, a peptide bond joins two amino acids and a water molecule is released. Following the hydrolysis reaction, the bond is broken due to the addition of water. Amino acids. Protein monomers. The name amino acid is appropriate because one of these groups is an NH2 amino group and the other is a COOH acid group. The third group is called an R group for the amino acid. Note that the central carbon atom in an amino acid bonds to the hydrogen atom and also to three other groups of the atoms, one which is called the R group, figure 3.15. Amino acids differ according to the particular R group, shaded in blue, figure 3.16. The R groups range in complexity from a single hydrogen atom to a complicated ring compound. Some R groups are polar and some are not. Also, the amino acid cysteine has an R group that ends with an SH group, which often serves as connect one chain of amino acids to another by disulfide bond. SS. Several other amino acids commonly found in cells are shown in figure 3.16. Each protein has a definite sequence of amino acids, and this leads to the levels of structure in a particular shape per protein. Figure 3.16, amino acids. Polypeptides contain 20 different kinds of amino acids, some of which are shown here. Amino acids differ by the particular R group, blue, attached to the central carbon. Some R groups are nonpolar and hydrophobic. Some are polar and hydrophilic. The ionized and hydrophilic. The amino acids are shown in the ionized form.
The shape of proteins. A protein can have up to 4 levels of structure, but not all proteins have all 4 levels. Primary structure. The primary structure of one protein is its own particular sequence of amino acids. The following analogy can help you see that hundreds of thousands of different polypeptides can be built from just 20 amino acids. The English alphabet contains only 26 letters, but almost an infinite number of words can be constructed by varying the number and sequence of these few letters. In the same way, many different proteins can result by varying in the number and sequence of just 20 amino acids. The secondary structure of a protein occurs when the polypeptides coil or folds in a particular way. Linus Pauling and Robert Corey, who began studying the structure of amino acids in the late 1930s, concluded that a coiling they called an alpha helix and a pleated sheet they called the beta sheet were two basic patterns of amino acids within a polypeptide. The names came from the fact that the alpha helix was the first pattern they discovered, and the beta sheet was the second pattern that they discovered. Hydrogen bonding often holds the secondary structure of a polypeptide in place. Hydrogen bonding between every fourth amino acid accounts for the spiral shape of the helix. In a beta sheet, the polypeptide turns back upon itself, and the hydrogen bonding occurs between extended links of the polypeptide. Fibrous proteins, which are structural proteins, exist as helices or pleated sheets that hydrogen bond to each other. Examples are keratin, a protein in hair and silk, a protein that forms spider webs. Both of these proteins have only a secondary structure. The tertiary structure is the folding that results in the final three-dimensional shape of a polypeptide. So-called globular proteins, which tend to ball up into rounded shapes, have a tertiary structure. Hydrogen bonds, ionic bonds, and covalent bonds between R groups all contribute to the tertiary structure of a polypeptide. Strong disulfide linkages in particular help maintain the tertiary shape. Hydrophobic R groups do not bond with other R groups, and they tend to collect in a common region where they are not exposed to water and can interact. Although hydrophobic interactions are not as strong as hydrogen bonds, they are important in creating and stabilizing the tertiary structure. Enzymes are globular proteins. Enzymes work best at a body temperature, and each one also has an optimal pH at which the rate of the reaction is highest. At this temperature and pH, the enzyme has its normal shape. A high temperature change in pH can disrupt the interactions that maintain the shape of the enzyme. When a protein loses its natural shape, it's said to be denatured. Some proteins have a quaternary structure because they consist of more than one polypeptide. Hemoglobin is a much-studied globular protein that consists of 4 polypeptides, and therefore it has a quaternary structure. Each polypeptide in hemoglobin has primary, secondary, and tertiary structure. Protein folding diseases. Proteins cannot function properly unless they fold into the correct shape. In recent years, it has been shown that the cell contains chaperone proteins, which help new proteins fold into their normal shape. At first, it seemed as if chaperone proteins ensured that the proteins folded properly, but now it seems they might correct any misfolding of a new protein. In any case, without fully functioning chaperone proteins, the cell's proteins may not be functional because they may have misfolded. Several diseases in humans, such as cystic fibrosis and Alzheimer's disease, are associated with misshapen proteins. The possibility exists that the diseases are due to missing or malfunctioning chaperone proteins. Other diseases in humans are due to misfolded proteins, but the cause may be different. For years, investigators have been studying fatal brain diseases, known as TSE, that have no cure because no infective agent can be found. Mad cow disease is a well-known example of a TSE disease. Now it appears that the TSE diseases could be due to misfolded proteins called prions that cause other proteins of the same type to fold the wrong way too. The possible relationship between prions and functioning chaperone proteins is now under investigation. Figure 3.17, Levels of Protein Organization. All proteins have a primary structure. Both fibrous and globular proteins have a secondary structure. They are either helixes, keratin or collagen, or pleated sheets, silk. Globular proteins always have a tertiary structure, and most have a quaternary structure. Examples, hemoglobin and enzymes such as RNAse. Figure 3.18, Fibrous proteins. Fibrous proteins are structural proteins. A. Keratin, found, for example, in hair, horns, and hooves, exemplifies fibrous proteins that are helical for most of their length. Keratin is a hydrogen-bonded triple helix. In this photo, Drew Barrymore has straight hair. B. In order to give her curly hair, water was used to disrupt the hydrogen bonds, and when the hair dried, new hydrogen bonding allowed it to take on the shape of a curler. A permanent wave lotion induces new covalent bonds within the helix. C. Silk made by spiders and silkworms exemplifies fibrous proteins that are pleated sheets for most of their length. Hydrogen bonding between parts of the molecule occurs as the pleated sheets double back on itself.
Check your progress. Question 1. The protein functions, metabolism, transport, and regulation in animals is shared by plants. Question 2. The primary structure of a protein is the protein sequence of amino acids that form a polypeptide.
A. The peptide bond includes a partially negative hydrogen. Hydrogen bonding between peptide bonds makes the protein secondary structure. B. Covalent bonding between R groups, such as disulfide linkages.